Hi everyone, welcome back. How are you all doing? I hope you are doing very well after the whole day of learning, school, lectures. So welcome back to the session 4 of Principles of Inheritance and Variation. So let's begin. I see that most of you are here. Kavya, Tasneem, Revati, Kartikeyan, Mirza. So all of you are here. Lakshmi, I know all of you. I don't know Nidarshana. I hope I am saying it right. Yeah. Hi Nidarshana, I am Nupur and I am taking these sessions for Principles of Inheritance and Variation, and this is session 4. So if you have seen the videos of the previous sessions, it's good. Otherwise, I think today's topic you will be able to follow. Otherwise, later just go back and see the videos whenever you have time. Okay. So let's begin. Overview of the chapter, we have seen it multiple times, right? We have discussed introduction, important terminologies, the 7 experimental characters that you have written down on a sheet of paper next to you, and you looked it up before the lecture along with the names of the scientists, okay? Then we saw the 3 Mendelian laws: dominance, segregation, independent assortment, deviations to Mendel's law, linkage, codominance, pleiotropy, incomplete dominance, and chromosomal theory of inheritance by Sutton and Bory, that genes are present on chromosomes. Okay, good. So, did you guys do the homework? Were you able to do the homework, guys? If you were able to do the homework, we will quickly start revising. Hi Lakshmi, hi Manish. I hope all of you are doing very well. So let's see how many of you were able to do the homework. Even if you were not able to do the homework, it's fine, okay? Just do it whenever you get time. Be regular, try a little bit to be regular, because this homework that I am giving you is not for 1 hour or 2 hours, okay? It's just for 5, 10, 15, maximum half an hour, not even that. And if you do it regularly with me, it will be very nice in the end. So just tell me if you were able to do the homework. Okay, and yesterday, in the chat, there was some question at some point, who discovered chromosomal mapping? And I said I will answer that later because I did not know. My sheet is not lying next to me that I had during my time. And generally, the most important scientists we always discuss during the lectures. Okay, apart from that, if you have curiosities, always ask. You can also look it up yourself, otherwise you can ask. So you can write this down. Alfred Henry Stuyvesant discovered chromosomal mapping. Hello Arvind, Shivani is here. So, were you able to understand this table, guys? This table, all those who were able to do the homework, very good. All those who could not find the time, this table is nothing but it is explaining you the behavior of chromosomes and genes during meiosis and fertilization and behavior of Mendel's factors. And now you know that Mendel's factors were nothing but genes. So basically both these columns say the same thing, and that is why this chromosomal theory of inheritance was done by Sutton and Bory. That genes are present on chromosomes. Good, so this table was clear, right? Chromosomes occur in pairs, Mendelian factors in pairs, they separate out, both of them separate out during segregation. One of them only passes to the gamete. You remember this, right? We did that from 2n during meiosis, it becomes n and n, which are the gametes. Okay, this takes place during meiosis. And meiosis takes place in the germ cells, okay, testis of the male and ova of the female, and during fertilization, it comes back again. So this is during fertilization. Okay, good. Then I believe that this table was clear. And then this question, were you able to solve this question? It was a little bit tricky, right? You had to think C, D, plus, plus. So, sometimes they can confuse you by giving you the names that you don't know, like C, D, plus, plus. But were you able to do the question? Were you able to get the solution, whether after seeing or without seeing? Did you understand the answer? Did you get the answer? Just let me know, and if you got the answer and if there is no doubt, then we can proceed from today's session. So Srivasthan is also here. I think I don't remember Srivasthan very well. If we are meeting for the first time, hi Srivasthan. And these are the sessions for principles of inheritance and variation, and whenever you get time, try to go through the previous sessions, okay? But you will be able to follow today's session. It's some different topic, so it will be fine. Just because there is a little lag, you know, maybe we have to be a little bit patient. Patience is the key. So I will wait till you, good. So I believe Srivasthan went through the sessions, very good. Good. So was this question clear? I can't see the replies somehow. I will refresh it. Okay. Okay, till you guys tell me it was fine or it was not fine, I cannot proceed, guys. Just tell me if you were able to solve this question. Okay, no problem. Good. See, you have to reply. Maybe I am getting the replies very late, but if you were not able to, it's okay. So see, we'll start. Individuals homozygous for CD genes. Yes, I will explain it. They were crossed with wild type. Okay, and then these were the 4 progenies. These are the 4 progenies. Yes, I will do. Okay, good. See, you have to tell me, right? That is why I told you that it is a little bit complicated question. Now you see, individual homozygous for CD gene were crossed with wild type. So CD was crossed with wild type. And the 4 different progenies that they have already told you were plus plus, CD, plus D and plus C. Now if you see, these are both what? They are the parental types. Why are they parental types? Because these are the parents, right? They are exactly similar to the parental types, and these 2 are the recombinants now. Okay, and they have asked you to find the distance between the genes C and D. And we know that percentage recombination is equal to that many map units. We did it yesterday, the chromosomal mapping, right? You remember we did this. The recombination frequency between gene 1 and 2, that is equal to the percentage of crossing over. Now what you have to do is you have to calculate the percentage of crossing over. And how will you do that? So if you look in the solution that was given here, the offsprings with parental combinations. And which are the 2 parental combinations? Plus plus and CD. These are the parental combinations. So these are 4 offsprings and these are exactly like the parents. They are 1750. It's a mathematics, you know, little bit mathematics. I'm very bad at mathematics, but this I can do. So offsprings now with non-parental combination. So these are the recombinants. Okay, so these are now the recombinants, and then total offsprings is a sum of all 4 of this, or the sum of parental combinants and non-parental combinants. Now you have to calculate the percentage of non-parental combination or the recombination frequency. See, that is why I told you this is a tricky question, and this is something new concept, that the percentage of non-parental combination will be number of recombinants in the offsprings divided by total number of offsprings into 100, which will give you 12%. What is 12%? 12% is the recombination frequency. And if you remember, I told you the percent of recombination frequency is exactly equal to the distance between the two genes. So the distance between the two genes is 12 map units. Is it clear? Let me know if it is clear. Yeah, I think today there is a lag a little bit more, but we will be patient, okay? So this was a mathematical question that was given in this continuity material, okay? The thing is, you have to prepare yourself in the highest standard. Generally, I haven't seen these questions in the previous year papers, which should not stop us from not doing them. Okay? Good, Kavya, you got it? Good. Others, just tell me if you all got it. Good. Perfect. Perfect. See, sometimes that is why, I asked you, because see, you had already looked at the question. You had realized, no, you cannot do it. That's why it is important to read. And then we discuss. We are always there for you. Okay. So then let's begin. Now is my time. You have to make me happy. This is something from session 1. Shrivastan, if you cannot answer this, it is okay. Try to, go through the previous sessions. For others, pure line breed refers to homozygosity, heterozygosity, linkage, both B and C. So what is the answer for this question? This is something, that I think we did in the first session. So let's see how many of you remember. So Tasneem answered. Lakshmi also answered. Kavya answered. Arvind, Mirza. Kartikeyan, are you there? See, I'm a strict teacher, no? Always checking. It's okay. Okay, guys, good. So you all remember. I'm happy. So with this happiness, let's start. So today's topic is sex determination. You see, we have already finished 6 major topics. Did you realize? Like, that's why, with genetics, we take just small portions every day and we do it. And today is sex determination. Let's begin. So methods of sex determination, there are 2, male heterogeneity, female heterogeneity. And heterogeneity means different sex gametes. Hetero generally, homo is same, hetero is different. We have done this before. Mono is also like one and same. So heterogeneity means different sex gametes. Male heterogeneity means different male sex gametes. Female heterogeneity means different female sex gametes. Okay, very simple as the name says. So let us look first at male heterogeneity and what are the methods of sex determination. It is XXXY type, and I am hoping that generally most of you know this diagram and I don't need to explain it much, because in humans we know we have 46 chromosomes, 23 pairs, 22 pairs of autosomes and 1 pair of sex chromosomes. Okay, so this is exactly the same. So 22 pairs of autosomes means 44 because human beings are diploid. So this 44 comes from the autosomes. Okay, and this is the sex chromosome. So this is the female here and this is the male here. Male heterogeneity means that the male will produce 2 different types of gametes. Very easy. You see here 44. So 44 will reduce to half, 22, meiosis. Meiosis 1, homologous chromosome separate, meiosis 2, sister chromatids separate. So 44 will become 22. So it is 22 into X and the other is 22 plus Y. Not into 22 plus X and 22 plus Y. So these are 2 different types of gametes. That is why male heterogeneity. And XXXY type is found in most of the fish, amphibians, reptiles and mammals. And this you have to remember. You can write it down on your cheat sheet, on the side if you want. But otherwise the examples, see, I always say concept, 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 because we are concept tree, right? We are the best. But we also have to memorize some things, and examples is one of these things. Name of the scientist is one of these things. The year is one of these things. Okay. So it is found in most of the fish, amphibians, reptiles and mammals. In such cases, the Y chromosome is male determining and absence of Y chromosome determines femaleness. Why? Because whenever there is a Y chromosome there are specific factors called as testis determining factor. And these testis determining factor result in the formation of testis in males, and whenever this Y chromosome is not present, it will be females. See, you already know it is XX in females and XY in males. That is nothing new for any of us. But the determinant is the Y chromosome. So what determines sex in amphibians? The presence of Y chromosome is male. Okay, and the absence of Y chromosome is female. Then the X containing and Y containing sperms can also be called as gymnosperm and androsperm respectively. Okay, so for example, the 22 plus X gamete and 22 plus Y gamete. This is what they are talking about, because sperms will contain half the material. Okay, after meiosis, sperms will contain only half of the genetic material. So these are the two different types of gametes that will be there. The second type of male heterogamety is XO and XX. XO is again male and XY is the female type, and again you have to remember the examples because they will ask you. For example, bugs, grasshoppers and locusts. Locusts are also, similar looking to grasshoppers. I just looked at before the lecture how they look. So they look like something like grasshoppers. So you can Google it later and see the image of locust, how they look. So bugs, grasshoppers and locusts, it is also male heterogamety, but now it is XO and XX. What does that mean? In such mechanism also the male is the heterogametic sex. Okay, again the sperms will be of two different types. 22 plus X, this will be one sperm. The other will be 22 plus 0. And why is it 0? 0, is it some special gene? No, 0 means nothing. So the only composition will be 22. But still it is male heterogamety because there are two different types of sperm gametes formed in the sperms. But in the ova, in the egg, it will be always the same, 22 plus X and 22 plus X. That is why it is also an example of male heterogamety. Okay. See, sometimes, the lectures will be like this. You have one thing after the other. Try to follow, because if you listen to this, something will go in your head. Even if you are tired, try to do like this or something like this. Wake yourself up. Okay. Good. So, I hope the main thing that you have to understand is what is male heterogamety when different types of sperms are produced. When the composition of sperm is different and when male is the sex determining factor. Now we see sex determination in Drosophila, and now you have to wake up because this is an important concept where they will ask you questions. Okay. So the diploid complement in Drosophila is 8, 6 plus 2 with 3 pairs of autosomes and 1 pair of sex chromosomes. Let's just compare what they say by this, The diploid complement in Drosophila is 8. In humans, the diploid component is 46. Okay, 6 plus 2 with 3 pairs of autosomes. In humans, we have 22 pairs of autosomes and 1 pair of sex chromosomes. So, always if you see so many numbers and you don't understand, just try to relate it with humans. Okay, because we are very clear with the human thing, right? 46 chromosomes, 23 pairs, 22 autosomes, 1 sex chromosome. So just try to do that. The sex mechanism in this case is XY and XX. But maleness is not decided by the presence of Y chromosome. Now what is this? We just discussed, no, that Y chromosome is the male determining factor in XXXY. We just discussed, and you'll be like, okay, another deviation, you know. So there is no deviation here. This is entirely a different sex determination concept. Here the sex is determined in an entirely different way from every other organism. Okay guys, why is this? Because the Y chromosome is inactive or inert. It doesn't matter if the Y chromosome is present or not, because all the genes on the Y chromosome are inactive. If you have studied, you know, molecular basis of inheritance, you know that the genes are inactive. They are heterochromatin. They are not going to be expressed. So that is why in case of Drosophila, it does not matter. Now is the time you have to take your pen and your sheet of the scientist, because according to the genic balance theory of Calvin-Bridges, write it down. I will wait for a couple of seconds. The genic balance theory was given by Calvin-Bridges, and it tells you how sex is determined in Drosophila. Please write it down, guys, please. It is very important. We have seen yesterday how a recent question from 2017 was there, right? Good. I'm hoping everybody of you wrote it down. Genic balance theory of Calvin-Bridges. The sex in Drosophila is determined by ratio of number of X chromosomes to the number of ploidy autosomes in the individual. Exactly how you will find it in books. This is exactly copy pasted from the books. So you have to memorize it. Okay? No, we'll try to understand it first. Okay? And then it will automatically be memorized. So ratio of number of X chromosomes to the number of autosomal sets. So let's go to the next slide. Okay, there is some, I'm sorry for this. Okay, good. So sex determination in Drosophila. It is achieved by a balance of female determinants on the X chromosome and male determinants on autosomes. Now you have to read these red things. Female determinants on X chromosome, which means the X chromosome will have the genes that will determine if it is a female, and the autosomes will have the genes that will determine if it is a male. Okay, so it will become very clear in a second. Give it some time. So normal flies have either one or two X chromosome. When it will be one X chromosome? When it is XY. When it will be two X chromosome? When it is XX. Okay, but you cannot say that XX is a female and XY is a male. Why? Because the male determinants are present on the autosomes and female determinants are present on the X chromosomes. So normal flies have either one or two X chromosomes. If they are diploid, they will have two sets of autosomes. What does sets of autosome means, guys? This is important to understand here. Sets of autosome means the ploidy number. Humans are diploid. We have two sets of chromosomes. Okay, there is a difference between set of chromosomes and pair of chromosomes. So basically set of chromosome means ploidy. Two sets of chromosomes are present in Drosophila. Why? Because they are diploid. Now write in the chat box. Okay, enough of me speaking. Now I want you to write number of how many sets of chromosomes are present in a tetraploid organism. 3n. I actually answered the question, but okay, let's see. How many sets of chromosomes are present in a tetraploid organism? What is the ploidy of a tetraploid organism? Anybody? Did you all just sleep? Or maybe there is a lag. Yes, 3 sets. Yes, there is a lag. I'm sorry. I know you are good students and you are trying very hard to listen. Yes. Okay, so you understand, right? How many sets of chromosomes are there in humans now? How many sets of chromosomes are there in humans? Next is now, how many sets of chromosomes are there in humans? Good. So humans have how many sets of chromosomes? There is a lag, but we are okay with it, right? We are cool people. We are patient people. So I don't know now if you're answering for the first or if you're answering for the second. In humans, how many sets of chromosomes do we have? Yes. Yes, perfect. So you have understood what is a set of chromosome. It is the ploidy level. Now, just forget about whatever I said about Drosophila till now. This will make everything clear, and I am hoping this will make everything clear. So you see, now what you have to do is, you have to take the ratio of number of X chromosomes to the sets of autosomes. So the number of X chromosome in this condition where it is XX is 2. And what is the set of autosomes in Drosophila normally? Normally Drosophilas are diploid, so 2 sets of chromosomes. Ratio of X is to A. Number of X chromosomes and sets of autosomes. It is 1, therefore it is a female. Next, in case of XY Drosophila, number of X chromosome is now 1, but the set of autosomes still remains 2 because it is a diploid Drosophila. The ratio is half. Now it is male. Okay, and why is it male? You see, here we said that it is achieved by a balance of male determinants on autosomes. Here, the number of autosomes is higher than the set of autosomes is higher than the number of X chromosomes, and the set of autosome determines maleness. Therefore, maleness characters are high. In case of female also it is 2, but here now the X chromosomes are also 2. So, there is a complete balance and therefore it is a female. Is it clear? It is a little bit complicated, but is it clear or should I repeat it? Tell me. It is number of X chromosome to the set of autosomes, and set of autosomes is nothing but it is ploidy. Drosophila is normally diploid. Okay, it is normally diploid and it has either XX or XY, but Y chromosome has nothing to do with it because it is inert. Nobody cares about the Y chromosome. What they care about is this ratio, whether it is 1, whether it is half. If it is 1, it is female. If it is half, it is male. Clear? Good. Good. Very good if it is clear. Chalo, then we proceed. Yes. So we have seen the male heterogeneity. We have seen the XX and XY condition. We have seen the XO and XX condition. And then we saw the third exception, that is Drosophila. So in which Y has no role to play. It is the ratio of number of X chromosome to the set of chromosomes. Now you are going to tell me one answer. There is a Drosophila in which this is the sex chromosome, but it has 4n ploidy. Normally Drosophila is 2n, but for example, let's say there is a condition, and these conditions are quite common in Drosophila. It becomes 4n. Now whether this will be a male or whether this will be a female, just write it down in the chat section. Good. So, Nidarshana, very good. See, I was wrong. Sometimes even I can be wrong. And you all got tricked, right? So, we have to all thank Nidarshana first because she told me that tetraploid is 4. It is diploid, it is triploid and tetraploid, and in my mind tetraploid was 3. I don't know why. So guys, I'm sorry. I'm really sorry for this. And I really thank Nidarshana for this. Tetraploid will be 4n. 3n is triploid. I'm really sorry for this. Sometimes it happens, when you are in the flow, you don't realize it. But Nidarshana, thanks very much for correcting me. I'm sorry, guys. Okay, coming back to this. So, I see male, 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 male. Perfect. You have got the concept. What is the ratio here? It is 2:4, which is half. Is it clear? So, that is why it is a male, although it has 2 X chromosomes. So, it is not about X chromosome and Y chromosome. It is about the number of X chromosomes to the ploidy level or to the set of autosomes. Okay. Good. And I'm really sorry again. So, we'll repeat it again. So, for a haploid organism, 1 set of chromosome. For a diploid organism, 2 sets of chromosome. For a triploid organism, 3 sets of chromosome. For tetraploid organism, 4 sets of chromosomes. Okay. I hope you forgive me. I'm sorry. Good. So, male heterogeneity is done. Now, let's go to female heterogeneity. So, this is nothing but ZZ and ZY system. And now, you have to again remember the examples. For example, birds, chick or fowl, few butterflies, moths. Okay, and what is in this case? In such case, male is the homogametic producing similar types of sperms. Okay, so earlier we saw that the male was producing different types of sperms, but now male is producing same types of sperms. Okay, for example, let's say 22 plus Z and 22 plus Z. Therefore, now female is heterogametic, producing 2 different types of ova. 22 plus Z and 22 plus Y. So, in these cases, female is the sex determining factor. Female is heterogametic because it is female heterogameity. And it is very easy because instead of XX, XY, now they have used ZZ and ZW. So, ZZ is for males and ZW is for females. So, unlike mammals, the sex in birds is decided by the ova or eggs of the females and not by the sperm of the males. Good. Is it clear? So, female heterogameity and male heterogameity, as the name suggests. Now, this is the diagram from your NCERT which will help us to revise everything. So, the first sex determination is XY-XX, which takes place in mammals, and here Y is the sex determining factor. If Y is present, it is male. If Y is absent, it is female. Next, here is Drosophila, which is again XX and XY, but the Y chromosome is inert. There is no role of the Y chromosome because all the genes on the Y chromosomes are inactivated, heterochromatized. They will not be expressed. Therefore, in Drosophila, it is number of X chromosome to the sets of autosomes. And tetraploid is 4 sets, not 3 sets. Okay. And then the third one is where the female is heterogametic. Okay. It takes place in chickens, birds, etc. So, it is ZW for females. 2 different types of gametes will be produced in females and female is the sex determining thing. In males, it is homogametic. So, I hope this part is clear. Is it clear? Okay. If it is clear, I will wait. I will wait till all of you tell me it is clear. So just let me know, because this is the end of this part. We still have one more part for this session. Good. Very good. Okay, then we solve a question. I will drink water and you will solve. See, this is again a previous year question. I haven't done it by myself. In Drosophila, the sex is determined by whether the egg is fertilized or develops parthenogenetically, the ratio of number of X chromosome to set of autosomes, X and Y chromosome, the ratio of pair of X chromosome to the pair of autosomes. Tell me the answer. I'm getting some answers. Very good. I'm just waiting for all of you to answer. Okay, good. I see most of you have answered, and I was expecting this, okay, that there will be a confusion between option number B and option number D. Okay, so some of you say option number B and some of you say option number D. So now let us try to understand the difference between sets of autosomes and pairs of autosomes, and again I will take the example of humans. What did I say? I said sets of autosomes is the ploidy. What are humans? Humans are diploid. Therefore, the sets of autosomes is 2. You know, that is why we represent always as 2n. Okay, because n is the haploid and the ploidy is 2n. And what are the pairs of autosomes? We have 22 pairs of autosomes. So, the sex is determined by number of X chromosome to the ploidy or to the set of autosomes. Now tell me, all those who answered the D option, they have retracted it. I can see that. But Manish is still saying D. Manish, is it clear? There is a difference between sets of autosomes and pairs of autosomes. Humans have 22 pairs of autosomes, but they have 2 sets of autosomes. Is it clear? See, these are the tricky questions. I know that this is a little bit tricky, but let's see. So, always try to understand these things by taking the examples of humans. We always say that a particular organism is haploid, a particular organism is diploid, a particular organism is tetraploid, triploid, but it has 22 pairs of chromosomes. Is it clear, guys? Are you able to understand what I'm trying to say, or is it just let me know? Yeah, I think today it's really late that I'm getting the responses, but it's okay. I will wait. I don't get any thumbs ups. Okay, Manish. Thank you. But I hope for others also it is clear. Is it clear? The lag makes me unhappy, because I'm not able to get if it is clear for you or not. Okay. Good. I start getting the thumbs ups now. I'm happy again. Good. So the right answer is option number B. Good. So next we do something that is sex chromatin, and again it is the time for to take your pen and write it down that M.L. Barr and Bertram in 1940 observed a darkly stained body inside the nucleus. You see this one here, close to the nuclear membrane in the cell body of neurons of the female cats. Okay, now we will read what exactly is sex chromatin and then I will try to explain you. As this was a darkly stained body and sex could be differentiated on the basis of presence or absence of such body, the body was called as sex chromatin body or Barr body. Now what happens, guys? Let's again come to humans. Okay, he observed in cats, but humans is always the easiest to understand. We know that females have the composition 22XX. This is females. Every cell of the female has this composition and every cell of the male has this composition. Okay, in each and every cell, in each and every nucleus of the body, it is 22 plus XX, 22 plus XY. Now you see here, females have 2 X chromosome and males have only 1 X chromosome. This is not right, right? We always talk about equality. Equality, you know. Women empowerment, we always want to be equal. So it is not fair if females have 2 X chromosomes. Therefore, one of this X chromosome is inactivated so that both females and males have equal number of X chromosomes, equality. Now next question you'll be like, no, but ma'am, males have Y chromosome. Why is that? We don't have Y chromosome. We should have something, females, you know. No. The Y chromosome is anyways a very small chromosome, and what that does is that basically has the factors that are required for the male reproductive organs, male accessory organs. So females don't need the Y chromosome at all. Therefore, one of the X chromosome is female is inactivated, because otherwise if we have 2 X chromosomes, we will have double the amount of all the genes, which is very bad. Okay, we will later see there are certain diseased conditions where it happens, but in a normal human female, one of the X chromosome is inactivated. Is it clear? So as this was a darkly stained body and sex could be differentiated on the basis of presence or absence of such body, the body was called sex chromatin body or Barr body, you know, and that is how it looked. And please write it down, M.L. Barr and Bertram, they observed the Barr body. That is why the name is Barr body, you know, to give importance to the scientists. We all are now repeating his name so many times, he must be happy from the top. Okay, so write it down. Is it clear? Is this concept of Barr body clear? Why one of the X chromosome is always inactivated in every cell of the human beings? And when I say every cell, I mean every somatic cell, not the germ cells. Okay, germ cells have to undergo meiosis. They have to make gametes. So there, while gametogenesis, it is active. These things will become more clear when we study, you know, mitosis, meiosis. But for now, every somatic cell, one of the X chromosome is inactivated because we are equal people. We want equality. Men and women are equal. Clear? Thumbs ups. Good. And later this body was found to be in the interphase nuclei of many cells of mammals. And this is important. The number of Barr bodies is always 1 less than the number of X chromosomes. There's always 1 less than the number of X chromosomes. Even when, you know, there are certain diseased conditions such as meta-female, meta-male, which we will see in the subsequent sessions. Even if, for example, this is one condition. Okay, for example, this is one condition. So this Barr body represents nothing but an inactivated X chromosome. For example, let's say this is the somatic cell. It has 22 plus XX. This will be, you know, a darkly stained body. And you know why it will be darkly stained? Because it is completely heterochromatized. It will contain this gene, this whole chromosome will contain a lot of proteins so that it is completely inactivated. It represents nothing but one of the inactivated X chromosome. Kavya, is it clear? Did I answer your question? This bar body is nothing but an inactivated X chromosome, and this is randomly, okay? Because see, when it is a female, it is XX. One X comes from the mother, one X comes from the father. Any of the X chromosome can be inactivated. Okay, there is no rule that one from the either from the mother, either from the father. Any of them can be inactivated. Is it clear? Bar body represents nothing but an inactivated X chromosome. What do you mean by inactivated? None of the genes will be transcribed. They will not be translated into proteins. Just like in Drosophila, you know, we said that the Y chromosome is inactive. But there it does not form a bar body. Here it forms a bar body, you know, that looks like this. And that is why it helps us to determine that, okay, this is a female. It also helps us to determine, okay, this is a female, and how we will see in the applications. Okay, now you have to tell me, the number of bar bodies is always 1 less than the number of X chromosome. Hypothetically, if this is the genotype of the sex chromosome, what are the number of bar bodies? Bar bodies is 1 less than the number of X chromosome. Tell me. What will be the number of bar bodies? X, Y. Let's say this is a hypothetical condition. Later in the subsequent sessions, we are also going to see such things. So what is the number of bar bodies? So bar bodies is 1 less than the number of X chromosomes. What is the number of X chromosomes here? 3. 3 minus 1, 2. 2 bar bodies. I see some of you have answered 2. All those who haven't answered or I haven't received because of the lag, I hope it is clear. Okay, I saw somebody at some point said 1, but okay, it is not 1. It is 1 less than the number of X chromosomes. Here the number of X chromosomes is 3. 3 minus 1, 2. You will see 2 bar bodies. In a normal female, always 1 bar body. In a normal male, no bar body. This is a normal male, right? 1 less than the number of X chromosomes. 1 X chromosome minus 1, 0. That is why in males there is no bar body. Very good. Everybody is answering 2. Very good, guys. Let's proceed. Applications of bar body. This is a slide that is flooded with sentences. It is just for you to understand. Okay, I will try to explain. Try to listen to me right now. Amniocentesis is a process by which you can determine the sex of the baby. What they do is they just take the amniotic fluid and then they determine the sex of the baby. And how? By bar body. They see the cell. If they find this black thing close to the nucleus, then it is a bar body and then it is a female. And if there is no bar body, then it is a male. Easy peasy, right? So it is a process by which amniotic fluid floating fetal cell is taken out from the pregnant mother around 6 to 10 weeks of pregnancy. And they are examined for the presence of bar body. Okay, amniocentesis, where you can determine the sex of the baby. You were always taught this, nobody told you how. By bar body. Okay. Then in forensic department, if you have seen, I don't know, when I was in India, I used to see a lot of CID, and if you have seen some of the other series, whenever there is a crime scene, there will be always this guy in a white coat collecting hair, collecting blood, collecting skin, nails, etc. What they can do? They can see the DNA, they can do fingerprinting, that we will again learn later, or they can see if there is a bar body or not. Because if they have 2 suspects, one male, one female, they can easily tell, which one it is by the presence or absence of bar body. If the bar body is present, then it is a female. Okay. And then in sports, sometimes what happens is people trick. It will be a male, but then it will show that it is a female to win the competition. Sometimes it happens, So at that time also they do these analysis for bar body. So these are the applications of bar body. Sometimes there can be questions, around this topic. Good. Then we are done with sex determination. And today's lecture is quite a lot, I feel. We'll quickly do mutations and mutagens. And see, in mutations and mutagens, there are very little new concepts, okay? Most of you already know all the concepts. So then what is mutation? The phenomena that results in alteration of DNA sequence and consequently result in changes in genotype and phenotype is called as mutation. But we all know what mutation is, right? For example, this is a DNA sequence. There will be some mutation here. But what is the problem when there is a mutation? The gene will get converted into a protein. This is important, guys, remember. If there is some changes on the DNA, it doesn't matter until and unless it comes out in the form of a protein. And this protein is then what will be altered. And this protein gives us the phenotype. Okay. For example, my skin is this color. But for example, there is no melanin production there. There is a mutation. There is no melanin production. I will be completely white. Okay, so that is the reason whenever there is a mutation on a gene, the problem occurs because the protein does not form properly. And then there are two types of mutation, gene mutation, chromosomal mutation. So let us first quickly look at the gene mutation, and I am hoping most of you know what is point mutation. What is point mutation? It involves change in a single nucleotide. Point, point generally you can say like 1 point. That is why single nucleotide. And it is a process in which new alleles of a gene are produced. Then there are two types. One is induced, one is spontaneous. How is it induced? Induced is externally, you induce something through certain agents called as mutagens. And spontaneous, it automatically occurs inside your body. One day your body just decides, I'm not in the mood today. I don't want to do a normal DNA sequence. Let me just do a mutated DNA sequence. No, that's not how it happens. If you remember your molecular basis of inheritance, whenever there is a DNA molecule and then there is DNA replication, there is this enzyme called as DNA polymerase. Okay, this DNA polymerase causes replication. But this DNA polymerase is not 100% efficient. And that is why, these point mutations can occur. And what is the problem now? See, there is a disease called as sickle cell anemia that we will discuss later in too much detail. Here what happens, the RBCs, they are sickle shape. That is why these people develop anemia. Now, what is the cause? The cause is a point mutation. You see, here it is GAG. So, look at the left side diagram first. Don't look at the right side. GAG gets transcribed to RNA. Normal protein is produced and the amino acid is glutamate. Okay. Then, when the mutation occurs, this A here is now converted to T in such a way that the mRNA sequence changes and now the protein has instead valine. You see, a single amino acid change can cause such a drastic change. That is what I meant when I said that this is the DNA and the mutation in the DNA will change the protein, and that is why you see a disease. So this is point mutation. The other type of mutation is frame shift mutation that we will discuss in molecular basis of inheritance, okay, in molecular biology. There are also recorded videos on the portal, on the channel, you can go through. Is this clear? Like this point mutation, I think you already knew this concept, but this example of sickle cell anemia and why this mutation occurs, DNA polymerase is not always 100% efficient. It can be lazy sometimes. Like we are also lazy. Generally on weekends we are very lazy. But you cannot be lazy, okay? You want to be doctors. Chalo, let's proceed. Next is chromosomal mutation. So this was gene mutation, just on the gene. And genes are present on chromosomes. Multiple genes are present on chromosome. Chromosomal theory of inheritance given by Sutton and Bory. You have the name written next to you. And I think I am repeating it so many times, it will get into your head, I promise. Just listen to me. Okay, so these chromosomal mutations can be due to change in the structure or due to change in the number. And what does that mean? We will look. So let's just talk about due to change in structure. So what are these structural changes? Deficiency, duplication. Exactly like the name says, Deficiency. Deficiency is always less of something. A, B, C, D, E, F, G, H. A, B, C, D, E, F. Oops. G, H is not there. Two of the genes are deficient. So that is why this is deficiency. Therefore, there will be change in the structure of this chromosome. Then it's duplication. A, B, C, D, E, F, G, H. A, B, C, D, E, A, B. You see, AB from here it is again now duplicated. So it is very easy as the name suggests. Deficiency, it is deficient. Duplication, it is duplicated. Clear? Exactly as the name suggests. Now comes very little bit tricky, inversion. A, B, C, D, E, F, G, H. This is the normal chromosome. Inversion A, B, D, C. Don't get confused why are they showing like this, because the sequence is fine. It is just inverted. Okay. You see here it is C and then it is D. Now it is D and here it is C. Also E is missing. Deficiency. If you did not get it, let's look at this. This is a normal gene sequence. 1, 2, 3, 4, 5, 6, 7, 8. Counting we all know very well. Now you see these two arrows. These are the points where inversion takes place. And that is why now it is changed. So now it is 1, 2, 6, 5, 4, 3, 7, 8. Is this inversion part clear? It happens nothing. There is this piece. It just gets converted like this. It's just from this to this. So is it clear? If it is clear, now this is the question for you here. This is the sequence, okay? It is not normal counting. 6, 1, 3, 5, 2, 4, 7. And you see these arrows. Here is where the inversion takes place. You have to tell me the new chromosomal sequence after inversion. If there are questions, you can ask. If I am going too fast, let me know. I know I am going a little bit fast, but I have a feeling these are the concepts that you already know. But if I am wrong, just write it down if I am going too fast. So just write down after inversion what will be the sequence. See, we are doing all these things because there will be questions. All these practicing and everything we are doing because there will be questions. That is the aim, to answer these questions. That is how we will clear the NEET exam. We will get a good rank, very good rank. So I will wait till you tell me what is the sequence of the inverted one. Okay. Perfect. Now I start getting the answers and this is the right answer. And all of you I think have given the right answer. Perfect guys. Very good. Very, very good. I am so happy. If I am going too fast, let me know. Good. And then the last one due to structural changes is reciprocal translocation. Okay. And what you have to remember in reciprocal translocation, it is always in non-homologous chromosomes, the chromosomes that are not related. It is a type of chromosome rearrangement involving exchange of chromosome segments between two chromosomes that do not belong to the same pair of chromosomes. Okay, because see, these are the pair of homologous chromosome. Even if there is something going on between these two, they can still handle it. But now look here. This is one chromosome, this is another different chromosome. Okay, they are not related. A, B, C, D, E, F, G, H, I, J, K, L, M, N, O, P. You see how the sequence is. This is sequence of one, this is sequence of the other. Now what is reciprocal translocation? A, B, C, D, E, F, G, P. We did not learn this, right? A, B, C, D, E, F, G, H. And here I, J, K, L, M, N, O, H, not P. What happened? There was translocation here. Okay, this piece got cut, came here. This piece got cut, came here. And this is now the new product is. You know, on a pictorial basis, it looks, okay, just one letter changed or just one gene changed in principle. But this is a very big problem. This is a problem that can lead to even cancer. Because if there is transfer of one gene, you know, that is a cancer causing gene, it can quickly cause a disease. So this is again another type of mutation due to structural changes of chromosomes. Clear, guys? So inversion and reciprocal translocation, is it clear? And see, here they have shown you an example, or I have shown you an example that has the same length, because not all chromosomes are equal in length. So sometimes it can happen, there is a big chromosome and then there is a small chromosome. And what happens is this big part and small part, and in the end, you know, what is the end result? So the one that was big will become small, the one that was small will become big, which is again a very big problem. Is it clear? Are these four clear? Just let me know. So deficiency, duplication, inversion, reciprocal translocation. Are these four concepts clear? Thumbs ups is what I'm waiting for. I know there is a lag, but it's perfect for us. We have survived 4 sessions together almost. This one is almost done. I know I am a little bit above time today, but I'm really sorry. Good, good. Good. I start seeing the thumbs ups now. Great. Very good. Good, good, good. Perfect. And then the other type of chromosomal mutation is due to change in number. When there is change in number of the sets of chromosomes, then it is called as polyploidy. And when there is change in number of pair of chromosomes, then it is called as aneuploidy. What is it? We have done sets, right? For example, human beings are 2n. Okay, we all know human beings are 2n. If there is polyploidy in human beings, the condition will be either triploid, 3n, 4n, and such conditions don't exist in humans. Those human beings are not born. If God forbid something like that happens, that human being is not born. Okay, but these are the conditions you will see a lot in flowers, and maybe in reproduction in flowering plants you have studied this. Okay, that the normal condition is 2n, but then it becomes 3n or 4n. Okay, so this is what polyploidy is. What is aneuploidy? Change in number of pair of chromosomes. For example, there are 23 pairs of chromosomes in human beings. So here what will happen? There will be change in number of pair of chromosomes. For example, let's say one of the pair is present in 3 copies. Okay, the rest is fine. 22 pairs are completely normal, but there is change in number of 1. And if this change is on chromosome number 22, 21, it is called as Down's syndrome. And we will see this, okay. What is polyploidy? What is aneuploidy? Polyploidy is when a normal organism, let's say, is 2n, it will become 3n. It will become 4n. It will become 5n. Such conditions are very common in plants. In humans, it doesn't exist. And aneuploidy is when there is change in number of a pair of chromosomes. Okay, like this. Normal are 22 pairs, one is changing. Is this clear? And tomorrow's class, we will see more examples of polyploidy and aneuploidy, and then it will become completely clear. Okay. So, is everything clear? Polyploidy, aneuploidy. Polyploidy is when there is change in complete set, From 2n it becomes 3n. Everything changes. Human beings are 2n. For example, they become 3n, 4n. All of them increases. And aneuploidy, change in number of pair of chromosomes. If there are 23 pairs, then only one pair will change. Is it good? So we are already little bit up time, but if you guys, okay, this we'll do tomorrow, promise. I will skip this. This is just the mutagens. And then we'll quickly revise. If everything is clear, shall I start revision? There are very few questions today. I'm sorry for the late and for the delay. But today was, see, we also have to finish the syllabus, right? As long as it is clear. If there is any doubt, again, we are there for you. Right here, right in the comment section. I will repeat it again, whenever. Good. Then let's revise. Oh, I already have the answer. Then we skip this. Good. Then let's start. Why do I have answers for all already? Okay, let's do this. I'm sorry, I don't know why I have answers for all this. Let's start with this question. In humans, and you see this question is asked in 2015 and also in 2017. So, in humans, the gamete contributed by the male determines whether the child produced will be male or female. And sex in humans is a polygenic trait depending upon cumulative effect of some genes on X chromosomes and some genes on Y chromosome. What is the answer? Another AIMS question. What is the answer? Just try to answer it and then we will see what is the answer. Assertion and reasoning type of question. In humans, the gametes contributed by the male determines whether the child produced will be male or female. Sex in humans is a polygenic trait depending on cumulative effect of some genes on X chromosome, some genes on Y chromosomes. Okay, some answers are coming. Okay. Anybody else wants to answer? Yeah, I think there is a lag. I don't like this lag, but we have to deal with it. It's okay. So, some of you answered A, B. Where are others? Why is not everybody answering? Read the assertion, read the reason. The right way to do this, read the assertion, see if it is true or false. Read the reason if it is true or false. Then see if reason is correct for the given assertion. And don't look at each other's answers, okay? Because I've seen that in the last lecture actually, when I was doing my homework and going through the video, some of you write the right answer and then you delete it because others are writing something else. So don't do that. Just give me your answer. Good, good. I see a lot of you have replied. Good. So let's go step by step. In humans, the gamete contributed by the male determines whether the child produced will be male or female. This is true because we said that in case of humans, Y will determine, because Y has this testis determining factor. Next is, sex in humans is a polygenic trait depending upon cumulative effect of some genes on X chromosomes and some genes on Y chromosomes. This is false. Okay guys, this is false. This is not true. It is not a cumulative effect. It is not a polygenic trait because only the Y chromosome, I told you, right? It is just the Y chromosome. Presence of Y chromosome in humans tells it's male, absence tells it's females. So this is false. You have to read, they're asking you about humans. Okay, so all those who answered A, if both assertion and reason are correct, and reason is the correct explanation of assertion. Some of you I see answered A. That is not correct. Why? Because sex in humans is not a polygenic trait and it is definitely not depending upon cumulative effect of some genes on X and some genes on Y. It is only and only on the Y chromosome. Presence of Y chromosome will result in testis determining factor, development of testis. Absence of Y chromosome will not lead to development of the testis. It will automatically lead to the female reproductive system. Is it clear? The right answer is C. For all those who answered A, Shivani, Ramesh, Srivatsan, Lakshmi. Guys, is it clear? Sex in humans is not a cumulative effect. Only and only the Y chromosome will determine that. Good. See, that is why, I know this was like a heavy lecture. It was not like our previous 3 lectures. And in the end also, we did not do all the questions, but we'll practice again. Also, the time is very up. Okay, good. So now we talk about the homework. So what is the homework for today? There is no homework today. Yay, because you have been such good students, for me. I decided not to give you homework, but I want to ask you to try to study half an hour more than what you study every day. Okay, see, when I was studying, when I was preparing for my exams, I am a morning person. I would eat my dinner by 9:30 and by 10 I would sleep. Then I would wake up at 4 in the morning. And around 8 was my school bus coming. So before I went to school, I already finished one chapter. And that was that chapter that I had studied a day before, in the lectures. You see, you are taking 3 lectures a day. So today, either in the night, and everybody has different schedule. My sister, she used to study till 3 in the night. Then she would wake up early, late, in the morning at whatever time. And it worked for both of us. So you have to find your way. When is your way of studying the best? And increase half an hour today. You can study anything. Study physics, chemistry, biology. You can study biotechnology, reproduction in flowering plants, genetics. Try to solve questions. You had physics lecture. Try to solve similar questions. Okay, will you try that? Half an hour more. Will you do that? I'll be very happy if you do that. Okay, and it's for you guys. You have to work hard. So that is the homework. Generally, you spend like 15-20 minutes for my homework every day, right? So this is what you have to do today. Because these concepts were quite theoretical. Okay, you will learn them eventually. And tomorrow we will do the remaining questions that we were not able to do today. Also because the time is a little bit more. I know you guys are hungry. But was the lecture fine? Was it interesting today? Were you able to get everything? Good. Very good. See, you want to be doctors, you have to work hard. You don't have an option. And this is the time, this is the time. You have time. You still have, if you are giving your NEET exam next year, you have time. If you are giving it in 2 years, you have time. But time will fly. Work hard every day. Okay, good then. I wish you a very nice evening. Enjoy your dinner. Try to study half an hour more, whether in the night or in the morning. Okay, bye, and I hope to see you all tomorrow. Same time, same place.